Hi, my name is Marjory Moreau, and today we are going to talk about PBPK modeling in chemical risk assessment. So pharmacokinetics, so PK, deals with the study of the absorption, distribution, metabolism, and elimination of drugs in the body. Pharmacokinetics helps us to understand how drugs are absorbed, distributed, metabolized, and eliminated in the body. This knowledge is essential in predicting the potential toxicity of drugs and assessing the risk of adverse effects. By studying the pharmacokinetic of a drug, toxicologists can identify the critical factors that affect its toxicity, such as the route of administration, the duration of exposure, and the metabolism and elimination of drug. So database compartmental models time course curves are usually analyzed by assuming particular model structures and estimating a small number of model parameters by curve fitting. So all processes are first order. Two problems arise from these database models. The first is saturation of elimination pathways, so models that are not first order, possibly that blood flow rather than metabolic capacity, for example, of an organ might limit clearance, or Cout from the central compartment does not increase as the metabolic capacity increases. That's another example. The other models, other than PK models, are PBPK models, so physiologically based pharmacokinetic models. Organ volumes and blood flows, metabolic and elimination rates are based on the physiology in PBPK models. They are mechanistically based with description of chemical movement using tissue blood flows. So PBPK models are a description of the body and processes within the body that affect the disposition of a chemical. Disposition or pharmacokinetics include the processes of absorption, distribution, metabolism, and excretion of chemicals. After development with necessary parameters and equations, the models calculate the concentration of the chemical or metabolites, if necessary, in different parts of the body using exposure concentrations as an input. So PBPK models are constructed as an interconnected system of compartments representing various tissues described by mass balance differential equations that are solved to predict the amount of chemical in each compartment over time. Usually when we develop a PBPK model, the process is always the same. So we identify a problem, we look at the literature for mechanism of toxicity to better understand the chemical of interest, biochemical constants, so we can parameterize the model with biochemical specific parameters, and we also look for physiological constant. And then we create our model and we run some simulation that we can compare to kinetic data in animals, because most of the data we have usually are in animals, except for drugs where we can find some human data. But most of the time we have animals data. And then after comparing to the kinetic data, we can see if the fitting with the in vivo data is good or not. If the fitting is not good, then we need to redesign and perhaps conduct critical experiments like in vitro experiment to have a better idea of the clearance of the chemical, for example, or the absorption. And we refine our model, and when the model has been refined, we run new simulations and compare again to the kinetic data. This time, if the calibration of the model is good, we can validate the model with another set of data. And then when the model has been validated and we've run some sensitivity and uncertainty analysis, we can extrapolate to humans. So when we build a PBPK model, again, like I've said previously, we need to define what compartments we need in the model. So the compartments that are needed really depend on the goal of the model. And also will depend on the metabolism and transport processes involved in the chemical analysis. For the parameters, physiology data like organ weight and blood flows can be found in the literature. Biochemical data like partition coefficient and metabolism can be predicted or derived from in vitro experiment. And then we develop the model equations. Like I said, it's a system of mass balance differential equations. And usually we have one equation for each tissue, and each tissue is connected with the blood flow. So here, for example, this equation, I'm not going to spend too much time because I know it's quite complicated. But this equation, for example, say that the rate of change of the amount of chemical in the liver, so AL for amount in liver, with respect to time, so dt, equal the blood flow times the arterial concentration, so blood flow here QL, times Ca, the arterial concentration, minus the blood flow leaving the tissue, so concentration divided by partition coefficient. What is leaving the tissue is the venous concentration, like I said, divided by the partition coefficient. After that, we have minus Vmax times Cl divided by Pl. So here, the Vmax is the maximum velocity times the liver concentration divided by the partition coefficient in the liver, which is the free concentration. And then we divide by the Michaelis constant Km plus the free concentration. So here you can see divided by Km, our Michaelis constant, plus the free concentration leaving the tissue. So this equation is just the equation for the liver, where we would need to add the oral dose coming in, but we have an equation similar to this one for each compartment. I'm not going to go too much through these slides because we've already talked about what needs to be considered in the model, the route of exposure, the different tissue, the metabolism, the excretion, etc. What is important to remember is in PBPK modeling, we need to keep the model simple, which is the principle of parsimony. We don't want to use more than is necessary. So what is the primary role of PBPK modeling? Well, it's basically to define the relationship between an external measure of exposure, so a dose, and an internal measure of exposure, so a concentration in a specific tissue or in blood, for example. So let's look at the applications of PBPK modeling. The first one is interspecies extrapolation, which means predicting the exposure for human based on animal. So here you have an example of interspecies extrapolation. So the first step is to parameterize an animal model, here rodents, define the internal dose metric. So we want to look at tissue-specific exposure, define a point of departure. So we need experimental data on exposure and adverse effect, and then run some simulations. So here we are going to run, for example, I don't know, oral simulations and look at a brain specific tissue concentration. And we are going to assume an equal risk at equivalent dose between rat and human. And then what we're going to do is reverse dosimetry. So we are going to assume that our brain concentration in the rat is equivalent to the human brain concentration. And then when we do reverse dosimetry, we are going to look at the human equivalent dose that will lead to the same concentration in brain than in the rat. Now, the second application is intra-species extrapolation, which means predicting exposure for vulnerable populations, such as children, the elderly, or occupational exposure population. So now, let's suppose that we know the external dose that corresponds to an adverse outcome in average humans. So we can run a simulation to calculate the relevant internal dose metric. So here we have an oral exposure of 1 milligram per kilogram a day that we run in our adult average human population model. And we find a dose metric, let's say it's blood concentration of 9 microgram per liter. So now we can use reverse dosimetry to determine the external dose that leads to the same internal dose metric in sensitive humans. This external dose is therefore expected to lead to the same adverse outcome in sensitive humans. So here we assume our internal dose metric to be the same than in the general population for the sensitive population, so 9 microgram per liter, which will lead to the same adverse effect. And we run an oral model, but for the sensitive population, and find what oral dose will lead to this concentration. And here in our example, for example, it's 0.7 milligram per kilogram a day. Before introducing the next application, it's important to do a reminder on uncertainty factors. So uncertainty factors are used to address the differences between the experimental data and the human situation. So for example, we have these uncertainty factors are the classic uncertainty factors. We have subchronic to chronic. So it's a 10 uncertainty factor. Low AL to no AL, it's an additional 10. When the database used to do the risk assessment is not complete, we can add another uncertainty factor of 10. And then we have a 100-fold uncertainty factor for interspecies differences and inter-individual differences. The use of PBPK models can allow us to reduce the overall uncertainty factors, allowing for more realistic exposure guidelines, which is the purpose in the advancement of the risk assessment process. So here it's an example of application of PBPK modeling with data-driven approaches for defining uncertainty factors in risk assessment. Usually, the US EPA, under the Food Quality Protection Act, or FQPA, apply some uncertainty factors to protect children. So these factors can go from 1 to 10. But does this default value adequate? Is this default value adequate, inadequate, or overly conservative for specific chemicals? So for the purpose of developing a safe dose level to the human population, including sensitive subgroups like children, regulators apply the safety factors to account for limitations in the data used and variability and uncertainty in the differences between animals and humans. To address the concern for age-related sensitivity to pyrethroids, life-stage physiologically based pharmacokinetic modeling supported by in vitro to in vivo extrapolation was conducted to predict age-dependent changes in target tissue exposure to pyrethroid. The purpose of this early-age PBPK model was to calculate a chemical-specific adjustment factor to address age-related pharmacokinetic differences for pyrethroids in humans. So that's one of the applications of PBPK modeling. And if you're interested into this application, I created a specific course about the pyrethroid case study that was developed for CAFRA using Pletham, because the PBPK model that was used to do the risk assessment that was submitted to the EPA in 2019, and where the EPA actually regulated using the PBPK model, and it was submitted in Plethem. So it was not a generic model like Plethem is, but it was a specific chemical model, but the interface was really similar to Plethem. So I invite you to look at the next course about the life stage PBPK modeling. So now, another application is route-to-route extrapolation, which is predicting concentration in blood or tissue from one route of exposure using data from another route of exposure. So if we know, for example, the oral dose that correspond to an adverse effect, we can run a simulation to calculate the relevant internal dose metric. And to apply route-to-route extrapolation, we can use reverse dosimetry to determine the inhaled concentration that leads again to the same internal dose metric. This inhaled concentration is therefore expected to lead to the same adverse effect. So it's exactly the same principle that the two previous examples that I gave. So another application is in vitro to in vivo extrapolation, or IVIVE. With the new toxicity paradigm and the increased use of NAMs, there has recently been greater recognition of the use of computational models like pharmacokinetic modeling and IVIVE to help interpret biological data and integrate various types of studies that can eventually be applied in a risk assessment context. IVIVE is really a valuable tool in extrapolating an in vitro point of departure from an in vitro toxicity assay to an equivalent applied dose in vivo. So basically, the chemical concentration active here in an experimental in vitro system are converted to equivalent levels in vivo by using pharmacokinetic model. There are some simple pharmacokinetic models used for IVIVE. They are really useful because they are simple. That's, for example, the model developed by Barbara Wetmore. They can be rapidly parameterized using in vitro measurements of clearance by hepatocytes and also plasma protein binding. But with more data, we can also use PBPK model to do IVIVE. Usually it's more chemical specific, but there's more and more models like HTTK or Plethem that can generate IVIV for large number of chemicals. So how can the simple PK models can be used? Well, they can be used to prioritize compounds. Here you have an example where you have thousands of chemicals here on the bottom, and here we have high throughput exposure predictions, probably from ExpoCast, and on the top we have the estimated dose to cause bioactivity coming from in vitro high throughput assays where we did high throughut IVIVE. And we can, when we have these two kind of data, we can calculate what we call an activity exposure ratio, or AER, which is kind of a margin of exposure. And we can see where the margin is extremely low. So we need to really look deeper into these chemicals. But usually there's a decreasing priority with an increasing margin of exposure. Another use of IVIVE is to use a PBPK model to do a more quantitative assessment of the exposure. So we can take the results of the in vitro assays. So it can be an AC50, for example, so a concentration in vitro. And using the PBPK model, we can estimate the daily exposure by doing reverse dosimetry. And we can do that by assuming that the concentration in the assay is equal to the blood concentration, but we can also make the assumption that the concentration is tissue specific. And for example, If we have a reproductive assay, we can assume that the target tissue might be the uterus, and so we can look at the concentration in the uterus as equivalent to the concentration in vitro and find out what's the equivalent daily exposure that would lead to the concentration in the target tissue. Let's look at an example. It's an example that will probably be familiar with the ACC members because it was published in I think in 2022. So 5 human-derived differentiated in vitro airway epithelial cell culture models were exposed to already known toxic vapor 1,3-DCP or 1,3-dichloropropene. We monitored the toxicity in these cultures 24 hours after acute exposure using an assay for transepithelial conductance, which is for to look at the epithelial barrier integrity, and also an assay for cytotoxicity. It was the LDH release, so lactate dehydrogenase release. Finally, we've developed an airway dosimetry computer model for 1,3-DCP vapor to predict in vivo external exposure scenarios that would reproduce toxic local tissue concentration as determined by in vitro experiment. Another application is reverse dosimetry. But first, before talking about reverse dosimetry, let's talk about exposure assessment. So traditionally, average daily exposure to environmental chemicals are estimated using epidemiological survey questionnaires, sometimes combined with measurements of environmental concentrations in order to inform on individuals' behavioral patterns and environmental conditions that may influence their exposure. But this traditional approach to exposure assessment leaves large uncertainties regarding actual realistic exposures. And part of these uncertainties are related to inter-individual variability in physiology, as well as the environment. So what's the source of exposure, what's the frequency of exposure, etc. So to reduce uncertainties, robust exposure assessments are needed. So to use biomonitoring data in understanding exposure, PBPK modeling can actually be used in a reverse dosimetry approach to assess a distribution of exposure possibly associated with specific blood or urine levels of compounds. Reverse dosimetry integrates PBPK modeling with exposure pattern characterization, Monte Carlo analysis, and statistical tools to estimate a distribution of exposure that are consistent with biomonitoring data in the population. If that is a subject that really interests you, I also created a specific course on reverse dosimetry. So here, what's the goal of probabilistic reverse dosimetry is to reconstruct the population exposure distribution. So when we do this reconstruction, we can derive a 50 percentile of exposure and also a 95 percentile, and compare with some reference dose that we have in the literature to derive a margin of exposure. Another application of PBPK modeling is the kinetically derived maximum dose, or KMD. KMD allows for design of in-life experiments that are not confounded by nonlinear effects, including saturation of absorption processes or metabolism. Something that is also really important for PBPK, I mean, it's not an application, but that's something that is really important, is the PBPK modeling report template. Evaluation of a model is essential, and model documentation is also essential for evaluation. So why do we need a PBPK modeling report template? Because we want to report the goal of the project, the development of the PBPK model, and how it will be used. There's different guidance already available in the literature. The EPA published one in 2020, FDA in Europe in 2018, they also developed guidelines on the reporting of PBPK modeling, or the EPCS in 2010. But there's a new guidance that has been published recently, and this new guidance is built on existing guidance on model application in risk assessment or good modeling practices from the US Environmental Agency, the World Health Organization, and the European Food Safety Authority. PBPK models are complex. And before being used confidently in decision-making context, it's really important that the model structure, parameters used, and performance of the model are evaluated. And as risk assessment is shifting from animal testing to new approaches based on in vitro and in silico methods, alternative strategies are needed to accurately integrate and use these non-animal data. And this guidance is really focusing on parameterizing and validating PBPK models when in vivo kinetic data are limited or not available, so especially for use in risk assessment. So it's just a reminder of things we've already talked about, but in a PBPK model, we have the model structure and the model parameters. And obviously, the biological basis of the structure and the model parameters is really important and needs to be reviewed. And for the model parameters, the reproducibility of the input parameters is important, as well as the sensitivity of output to input. But the model structure and the parameters are not the only thing that make the PBPK model. The mathematical equations that are relating all the compartments together through the blood flow is also really important, and fit and predictivity on those metrics are something that needs to be evaluated. So this report template is similar to reporting formats developed in other guidance, and the template will be compiled by the model developer to report PBPK modeling analysis in the context of specific regulatory assessment. The model performance is really important. I've went to different conferences lately where there's always this question of how do we define if a model is good enough? I've always worked with the guideline from IPCS, which means that in PBPK modeling, predictions that are on average within a factor of 2 of the experimental data are considered adequate. Other people, well, it's also the same for the US EPA. It's a factor of 2. So, for example, these simulations here are considered as really good. But lately, I've heard more people saying that actually a factor of 3 is also considered as adequate. And the explanation is that usually the in vivo data that we are using to calibrate and validate our models also have a lot of uncertainty. So that's why having a factor of 3 is also adequate. So at Testovation, we've developed different set of tools these past few years. Obviously, PLATHEM is the main one, which is the population life course exposure to health effects model. But we also derived a module for doing high throughput IVIVE, reverse dosimetry, and also KMD, which is not called KMD anymore. It's called DPA, which is dose proportionality analysis tool. So let's take a look at the recent progress we've made in all these user-friendly tools. So first, the dose proportionality analysis tool. We've created and upgraded this tool with the EPA. The goal is to develop a case study that we can publish, hopefully by the end of the year. But here you have the link to have access to the tool, the DPA tool. And basically, it's really simple. It's really similar to Plethem. You have the physiological tab where you can input all the parameters for rat or human. In the chemical specific tab, you can put your chemical specific parameters, and then you can run some simulations. So here you can select the route of exposure that you want, and you have the choice between IV, inhalation, oral, and oral feeding. For rat, obviously, not for human. You can select the time of exposure, the length of exposure. You can select if you want some Monte Carlo simulation or not, and also if you want time course analysis or dose response analysis. There's also a tab to do some optimizations. So for example, if you have some in vivo data and you want to fit the simulation to the in vivo data, you can use the optimization tab to do so. So for the new version of Plethem, there is a package that might be available in the future on CRAN, but we realized that sometimes it's not really easy for everybody to download these kind of packages. So we are working right now on an online version that will be available before the end of the year. So that's the forward dosimetry interface. So you can select the module that you want. For now, there's just 2 or 3 modules. So you have the DPA module that we were working on with DPA. You have also the classic generic model for Plethem. But with time, the goal is to add other modules like a library of PBPK modules available. You can select the species of interest and the chemical of interest. And then you can select the tab ADME, where you can put the chemical specific parameters. You have a physiological tab, the exposure tab, and then you can run some simulation. So here in the Run Simulation tab, there's a few new things where you can create a username and a run name for your simulation, so you can save them and go back to them in the future. You can also run forward dosimetry and Monte Carlo simulations. Now for the reverse dosimetry interface. So reverse dosimetry integrates PBPK modeling with exposure pattern characterization, Monte Carlo analysis, and statistical tools to estimate a distribution of exposures that are consistent with biomonitoring data in a population. So we created a specific interface where you can upload Monte Carlo simulation from any PBPK model. You can also upload your population concentration data, so your biomonitoring data from NHANES or any other source. And you can run your sensitivity analysis. Here you have the results tab, where you will be able to see your CDF, so your cumulative distribution function, and a table with your percentile for the distribution of exposure in the population. This is the online version. So this reverse dosimetry is available through Plethem in CRAN, but there's also a specific online version. Here it's the link. And the first page gives you access to a small course on reverse dosimetry and how to use the application. And then after that, it's really easy to use. So that was all the application we've developed at Cytovation. Thank you for listening at this course. And if you have any question, again, my name is Marjory Moreau. I'm the Associate Director of the CompTox division. And don't hesitate to reach out to me if you want to talk about PBPK modeling or have any question about this presentation. Thank you.